Hello, in this video we're going to be talking about the genetic code. Let's not waste any of your time. Let's get right to it. Now, when we're talking genetic code, what we want to think about is, again, we're thinking genes, okay? But what we're thinking about is the process of making proteins. Now, one of the things that we're going to look at this diagram that's here beside me. You'll notice that you can see a kind of a series of the circles are amino acids, and then below that is the RNA strand that is being used to form that string of amino acids. And each one of those, you can see kind of alternating colors of light and dark green, sets of three. Okay, a set of three nucleotides on an RNA strand is called a codon, a group of three. And so each set of three then determines which amino acid is selected next and added to the string. And of course, based on what the string is made up of, determines what protein you make. That's kind of the idea of the genetic code. One of the things that Crick and Sydney, as kind of working together, they learned that which nucleotides code for which amino acids, and of course they made a big chart which we're about to see, and hopefully you've seen this chart before. There's lots of versions of it. One we're going to look at is pretty straightforward. It just has a list of the potentials and what amino acid they string for. Now, something else that they did was to try to understand how mutations, which can be in a lot of ways, mutations can be just simply removals, additions, but it's just simply a change in what's going on. And if you look at the diagram beside me, at the top part is they take out one nucleotide, and when that one nucleotide is taken out, you can see that it changes, okay, the amino acids that are selected. In other words, then you're going to change the entire protein. So if you have a specific protein that is being made by a particular RNA strand, and it's an important protein, and it gets a mutation, and now it's no longer making that protein, then that's going to obviously have some effects later on down the line. So the take home here is, if you just take out one, and essentially you're just going to move all the other letters down, and if you're working with sets of three and you change those letters, then you are simply going to make you're going to select for a different amino acid, which will make a different protein. The one below, on the bottom half of that diagram, is if you take out an entire set of three. In other words, you just take out a set of three and then move all the other blocks down the line. What you can see is that after the deletion, then the same amino acids are selected for, because you haven't messed up the order of three. So if you have an RNA strand that's making for a particular protein, and the mutation happens early, okay, then maybe, or I should say, how do I want to say that? I'll just go with early, like I said, early. Then all the other amino acids behind it are the same. And if it happens late, then you have a kind of a stop in the string, and then you have the remaining. So it depends on where it happens, what the effect might be. So it could be minimized just based on that position, but certainly what happens before and what happens after is the same, whereas when you take out just one, everything after is completely different, because it's important the thing to think about is it's important that each frame, each set of 3 is read properly. And if you change one of those, you are going to potentially change the protein that is being made. Now here's that chart that I was referring to, and this one's kind of small. This will be in your PowerPoint, and you can certainly Google genetic code amino acid table and look at one closer if it's too small. But Marshall Newberg played a big part in also helping identify which amino acids each codon codes for. And you can see on the chart, even if you can't read it because it's too small, you can see based on color coding how it's set up. There are some amino acids that are coded for by multiple codons, and essentially what that simply means is that the first 2 letters are the same, and then the last letter could be any one of the 4 possibilities, A, U, C, or G, but it's going to get the same amino acid. So what that tells you is that the 3rd digit isn't always important, but the first digit is super important because that tells you which amino acid you're gonna be potentially getting. And you can think about that kind of like a library. If you want fiction, but they take you to the nonfiction section, then you're going to get an entirely different book than what you were thinking about. But if you wanted fiction by a particular author, but they took you to the fiction to a different author, you could get the same kind of book, just not quite the one you were looking for. So that'd be like if the difference in the letters was in the middle, okay. And then if you wanted fiction by a particular author or a particular book by a particular author, all 3 variables, and they took you to fiction and to that author, but not quite to that book, then again, that'd be like if the change was in the 3rd variable. So the further into the codon the change is, the greater the change is going to be. Now, some other things to think about when we get in, and this will make more sense when we get into transcription and translation, but there are codons that are associated with stopping the reading of the RNA strand, and there are codons that are associated with starting. Starting the reading of the RNA strand. And you can see on the screen what those are. And because particular codons can select for the same amino acid, that's called degenerate, and that just simply means that a particular amino acid can be selected for by multiple codons, but they typically always start with the same first letter. So if you look at that screen over there, if you see an amino acid circle and it's in a box by itself, and then there's 4 of the lighter purplish things, that means that any of the 4 can, but there are some that in a box that there's 2. That means the first 2 letters are the same, and then the last digit is separated. Maybe A and U codes for this amino acid, and C and G code for that amino acid. And there are some, like in the very far right-hand upper corner, where there are 3 different things that are coded for amongst the 4 amino acids. So it varies greatly, but again, we'll look at one of these in class, up close and personal. And there was one more to say, but I couldn't squeeze it onto the previous slide. The idea that all organisms use this process means that kind of early on in the development process, whatever your mindset is on how that works, church or science, that it's pretty universal that it all kind of works the same, which points to kind of a common ancestry type situation. Well, that's our video over the genetic code. When we come back next time, we're going to be looking at transcription. So we'll see you then.